Practical Issues, Paper 3, Issues and Debates. Practical issues try to ensure replicability and to increase reliability by trying to control research. Practical issues impact choices researchers make when selecting measures in their research to meet the aims. For example, gathering a sample, methods, cost and implication, the use of animals, the difficulty to control extraneous variables, matching participants, independent group designs, and volunteer sampling. I will elaborate further on this later in the video. Validity. Practical issues do try to increase validity. When you do have a practical issue, it decreases validity. Validity versus ethics. This is when researchers do not reveal their true aims to reduce demand characteristics. For example, Milgram did not tell participants that this was a test on obedience. Social desirability. To design a questionnaire and interview that limits participants' wish to look good. Measurements. Researchers must make sure to measure results in a meaningful/valid way. E.G. PET scans. PET scans are valid because they're scientific and should be non-biased. However, you have to interpret PET scans. You have to interpret what is blue and what is red. Therefore, it can be subjective to bias, and therefore, it is not valid. Controls. Researchers try to minimize the influence of confounding variables by using controls. For example, Badley had the same rate of presentation, 3 seconds. Ecological validity. Settings and tasks of a study may allow results to be applied to other situations. For example, Sheriff was a naturalistic study. It was done in a camp, and Watson and Rayner was a lab experiment. Therefore, Sheriff can be applied to everyday life, whereas Watson and Rayner can't because it was a laboratory experiment. Reliability and standardization. Standardization directly increases the reliability by keeping procedures the same and the same between participants. For example, Milgram had the same procedure between participants. Reliability over time. Test-retest measure of reliability detects whether a measurement in studies or a diagnosis in psychology is consistent from one occasion to another. For example, Milgram's variation shows that it's consistent, and so does his replications like Berger. Reliability between researchers. 2 researchers or more assessing data. Both Schmolk and Sheriff had interrelated reliability because they had 2 researchers assessing the data. The different types of sampling that can affect practical issues. For example, convenience sampling. This is a non-probability sampling method where data is collected easily and accessibly and from available groups of people where you will have to recruit participants for your study. A subject pool is an established group of people who have agreed to be contacted about participating in research studies. For example, Brendigan contacted a sample pool to get his twins. Researchers post descriptions about their studies online and participants will sign up to participate. Participants who are not in the subject pool can also be recruited by this type of publicising. During field experiments, the task is not to recruit participants but to select them. It's important that this kind of selection is done according to a well-defined set of rules that are established before the data begins and that can be clearly defined afterwards. For example, when Berger carried out their study, they wanted non-psychology student participants and they wanted students with non-medical conditions that could be impacted during the study. These are their rules that was clearly set out before they selected participants. Finally, a volunteer sample is a non-probability technique where participants choose to take part in the study. Some participants may receive a form of compensation for being part of the studies, however they're still a volunteer. Volunteers can be better than non-volunteers. This is because they're more interested in the topic, they're more educated, they have greater need for approval, they have higher intelligence, they're more sociable, and they are in a higher social class normally. However, this leads to an issue of external validity, where these characteristics are not normal and they're not applicable to the general population, so therefore these studies cannot be applied to real life. To standardize a procedure, you need to control the extraneous and confounding variables. Extraneous variable is any unwanted factor in a study, other than the independent variable that could influence a result, bracket the dependent variable, making it hard to know if the independent variable is truly the cause of the effect. A confounding variable, however, is an unplanned third factor that affects the both independent variable and the dependent variable creating a false, misleading cause and effect relationship, ruining the external validity by confusing the true I and D effect. Extraneous variables affect participants' behaviour. They make the independent variable more difficult to detect. They systematically vary across conditions, and they become confounding variables and provide alternative explanations for the results. One reason for extraneous variables is when there's multiple experimenters. For example, Watson and Rayner had two experimenters, Watson and Rayner. As well as this, the experimenter's sex and the participant's sex can affect the results of the study. This is an extraneous variable. To try to control this, and I will elaborate further later, we can do a blind condition. This can be double blind or single blind. Controlling extraneous variables. This shows how to control extraneous variables in clinical psychology. However, you can also do this in laboratory experiments. Randomized clinical trials. These determine whether a treatment works. Participants are randomly assigned to either conditions, in which they either receive a treatment or a control condition where they do not receive the treatment. If participants are in the treatment condition, they end up better off than the participants in the control condition. This means that the treatment works. In the no treatment condition, participants receive no treatments. However, this can lead to the placebo effect. A placebo effect is when participants experience a real improvement even though they aren't getting a treatment, they're getting a fake treatment. Many participants who have been in the control condition are offered the opportunity to have the real treatment once it's revealed that they are in the no treatment condition. For example, Potts did this. A solution to the placebo effect is to leave out the control condition and to compare the treatment to any new treatments to see the best option, so therefore you can tell if your treatment is worth going on with. Finally, a waitlist control condition. You have participants who are waiting to have the treatment. However, this allows the researcher to control the participants who are waiting to receive the treatment to the participants who are currently receiving it, so you can see if they are improving or not. Experimenters should record demographic information. This is the date, the time, the place of testing, and the experimenter who did the testing. As well as this, they should write down any unusual occurrences or questions that come up. This information can be used later when analysing the data or if questions arises about a particular participant or testing session. Participants' identities should be kept confidential. Their names and any other identifying information should not be included in the data. To identify participants, they should be assigned an identification number to each participant. This is usually sufficient, or a pseudo name is used. For example, Baby Albert is not Baby Albert's real name. They use a pseudo name. A manipulation check. A manipulation check is where the researcher tries to confirm that the independent variable was successfully manipulated. A pilot study. A pilot study is a small-scale study conducted to make sure that the new procedure works as planned. There are several important questions that can be answered by conducting a pilot test. Do participants understand the instructions? What kind of misunderstandings do participants have? What kind of mistakes do they make? And what kind of questions do they ask? Do participants become bored or frustrated? Is an indirect manipulation effective? Can participants guess the research question or hypothesis? How long does the procedure take? Are computer programs or other automated procedures working properly? Are data being recorded effectively? During a pilot study, experimenters observe participants carefully during the procedure and talk to them afterwards. Participants are often hesitant to criticize a study in front of a researcher. However, the researcher must comfort them and help them to understand that they are generally interested in their feedback, as their feedback will help them to improve the procedure. If the procedure works as planned, then the researcher can continue with the study. If there are problems to be solved, then the researcher will solve them, create a new procedure, do a new pilot study, and then continue with this process until they're ready to proceed to the actual study. I will be going through your AO3, however, I will assume that you know the studies, so I will be going through it very shortly and not in detail. Practical issues in social psychology. Demand characteristics in obedience (prejudice). Sheriff. Later interviews revealed that the boys knew that they were being recorded and knew that the staff were taking notes. This can lead to demand characteristics. Social desirability bias, used by participants to mask prejudice. Prejudice is too subtle to be detected by a questionnaire. However, Katzen and Braley conducted a questionnaire to investigate national stereotypes of Americans and about other cultures. The majority of students classed African Americans as superstitious, and questionnaires are conducted to check understanding, using opportunity sampling in our practical investigations, as we did not have access to other year groups. Sheriff. They used a convenience sample of 11 to 12 middle-class boys. They had a high level of controls as the staff were researchers, but the boys saw cameras. This led to demand characteristics. Berger used a convenience sample, but it was screened. It was controlled to see if they were psychology students and if they had a condition that may affect them in the study. It was a highly standardised procedure, and it was a reliable procedure because it was Milgram's procedure that has been replicated again and again and again. Milgram. It was replicated in other conditions to ensure obedience was down to the authority figure. This controls extraneous variables. It was a convenience sample, though, of only practical issues within biological psychology. Some activity patterns may occur due to chance. If brain scanning method is flawed, then evidence isn't reliable. Activity may be in small parts of layers of activity patterns. Brain scannings are objective measures, but some say they are flawed and don't measure what they are intended to. Difficulty to measure transmission of neurotransmitters, as it's not easily accessible. Rain used a subject pool of NGRIs. However, they were matched on age, gender, and ethnicity. There was no pilot study that was conducted. He used PET scans, and it can be unclear and misinterpreted, which can lead to bias. As well as this, Rain used animal research to back up their claims and as background research, which is not reliable, as the extrapolation as animals to a human brain is not reliable. Their brains are a lot smaller than ours, and they cannot think about things like morality. However, mice do share 70% of the same brain matter that we do. Brendigan used a convenience/subject pool. They used leftovers participants, however it had to be MZ/DS twins. A variable that was not controlled during their study was the language barrier. Practical issues within learning psychology. Bandora's studies of modelling took place within an artificial setting, so it lacks ecological validity. Observing behaviour can be subjective. It involves learning about behaviours through animals, children, so it's difficult to generalise to humans. Humans have self-awareness, so they can show demand characteristics during experiments. Pavlov repeated similar experiments numerous times with multiple dogs, using controls, therefore it's reliable. Pavlov used a standardised procedure, repetition of stimulus, and a controlled amount of saliva production. It had little tubes in the dog's mouth to see how much saliva was produced. They also tested a hypothesis, thinking it was first the lab coat, then decided it was a bell and food association. Watson and Rayner. They used a convenient sample. They knew the mother of the baby, of a healthy baby, and used that one baby. They controlled different places, and they had control over what stimuli was used. Bastian used a control sample of mostly women. It was biased. They tried to control factors like self-humanity. Bandora. They controlled the reward and the TV. They had a control condition where participants were just put in the room and played. Participants was a convenient sample as well of nursery-school children. Cognitive psychology. I'm only going to talk about the experiments. Badley used 72 British volunteer psychology students, which means that they're better and more knowledgeable about the topic. He controlled variables like rate of word presenting and time of recall, which were all 3 seconds, and this means it had iterated reliability. Smoke. They had a subject pool of 3 medial temporal lobe and interval temporal cortex, 2 hippocampus, HM, and matched these participants with 6 participants for age, sex, and education. Schmock used a standardised procedure which can be replicated today. However, one reason why Schmock could not be replicated is because of his subject pool. It is so rare. Psychology. Clinical psychology focuses on qualitative data, which is difficult to analyse and is subject to interpretation, so therefore the conclusions can not be reliable. Carlson et al. The Carlson et al. Could have researcher bias due to wanting different results. Rosenhan had many variables like the symptoms of the pseudo patients, the IV, and the diagnosis of the patients being the DV. He used an observational method. Rosenhan was one of the participants. This could have been biased and skewed the results. Also, it was self-report. POTS. Convenient/volunteer sample of 112 children. They did a baseline and they had a placebo so they could see the difference. This study is the study where you can go back to the controlling extraneous variable slides and you can literally use that slide and then use POTS. Clinical psychology. Now this is the optional unit, so this may not apply to you. However, Lewis and Palmer, they used a convenient sample of uni students. It was a very standardized procedure. They controlled the clip's order, they used the same clip for all, and they isolated the effect of leading questions. Bradbury and Williams, they had several controls. They had the quality of evidence, the strength of the case, the length of the trials, the length of the jury deliberations, the presence of written instructions to the jury, the case type, and the lawyer type. It was a very controlled and very highly reliable study. Finally, practical issues involving animals. Animals should be selected with consideration to how similar they are to humans. Cost. Animals don't have to be paid, unlike humans. However, the cost of buying lab animals from home office and the cost of feeding them and keeping them is expensive. Danger. Animals can bite, scratch, and even carry diseases. This is dangerous to the human researchers. Space. Animals are typically small and take up less space than humans. However, they are also given suitable space for their needs and the needs of the species, so that they can maintain the territory and feel secure. Sometimes a whole room needs to be set aside for animals. Supervision. Researchers can go home on the weekend, and human participants have time off, but animals need to be cared for continuously, so someone has to visit them, feed them, water them, and possibly release them and interact with them for social time. Pavlov used dogs to show how salivation response can be caused by a conditioned stimulus. Andy and Vetemali stimulated the hypothalamus and basal ganglia of cats. The animals had aggressive seizures. Differences between humans and animals means studies like this lack generalizability and validity. Applications. Morning sickness in drugs show negative outcomes for humans but not for dogs. Multiple generations of animals can be studied quickly as they reproduce quickly. Drugs have been developed from animal research which has benefited both animals and humans, e.g. insulin for diabetics. I've only talked about Pavlov and Andy and Valimarty. However, you can talk about Skinner, you can talk about Ferrari, you can talk about so many different studies that use animals and just evaluate them. I'd just like to remind you that your AO1 is what are practical issues and your AO3 is applying them to a study like Berger, Milgram, Schmolk, HM, things like that. And good luck!